Okay, I'm good to go.
Okay, then
Okay.
Sorry, didn't realize I was on mute.
Carolyn, the floor is all yours.
Okay, great. My name's Carolyn Straub, and I am a second-year graduate student in the Department of Pharmaceutical Sciences, and I'm studying medicinal chemistry. And I am excited to share with you my presentation today on the anti-addictive properties and potential pharmacological significance of selective alpha-3 beta-4 nicotinic acetylcholine receptor antagonists. So there are many channels in the brain that allow ions to move in and out of cells, and one of these channels is called a nicotinic acetylcholine receptor. So nicotinic acetylcholine receptors are a class of ligand-gated ion channel that allow the passage of cations, a net passage of cations into the cell, and can be regulated through the binding of small molecules, such as acetylcholine, which is the endogenous ligand agonist of nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors participate in a variety of really important central nervous system functions, such as excitability and neurotransmitter release. And in the brain, there are 8 different nicotinic acetylcholine receptor subunits, at least that we know of to exist. And these subunits can combine to form multiple, very functionally diverse, both homo and hetero pentameric complexes. Here, I'll show you my pointer. So here is an example of 2 here. These are both hetero pentameric complexes, and these are the 2 that I'm going to be focusing on in the presentation, alpha-3 beta-4 and alpha-4 beta-2. So this is because alpha-4 beta-2 is the most commonly found in the CNS, while alpha-3 beta-4, I'm going to share with you why it is relevant for us. Recent studies suggest that alpha-3 beta-4 nicotinic acetylcholine receptors are implicated in substance abuse and addiction in rodents. So, for example, selective antagonists of alpha-3 beta-4 are known to reduce the self-administration of stimulants, block the rewarding effects of stimulants, and reduce the presence of withdrawal symptoms. However, investigation into the unique role of alpha-3 beta-4 is currently limited because there's a lack of subtype selective small molecules at our disposal. Because alpha-3 beta-4 and alpha-4 beta-2, the 2 subtypes that I mentioned previously, they have different physiological roles. It would be really helpful if we could develop small molecules that only interact with one or the other, but not both. So if such a small molecule existed, it could be used as a tool to study the physiological roles of each subtype, particularly in addiction, as well as learn about the properties of small molecules that cause them to have this selectivity. So the development of a potent and selective alpha-3 beta-4 antagonist would allow the specific physiological role of alpha-3 beta-4 to be investigated, as well as the characteristics of ligands that impart selectivity. Although many Americans are aware of the ongoing opioid crisis, it is a surprising statistic to many that 27% of synthetic opioid-related overdose deaths in 2016 involved psychostimulants. This signals that we need to address psychostimulant abuse as part of a comprehensive response to the opioid epidemic. So despite this need for suitable anti-addictive treatments, there are currently no FDA-approved drugs for addressing psychostimulant addiction. Alpha-3 beta-4 antagonists play an important role in solving this problem, potentially, because they've already been shown to decrease drug-seeking behavior in stimulant-addicted rodents. Which I shared with you on the previous slide. And understanding more about alpha-3 beta-4's role in psychostimulant addiction would move us all forward towards the development of the first FDA-approved treatment for psychostimulant addiction, which would make a huge impact on our community and those of us in our community struggling with psychostimulant addiction. So today I'm going to share with you in depth more about 3 alpha-3 beta-4 antagonists, where their selectivity lies and their pharmacological significance. So the first that I'll focus on with you today is Ibogaine. So Ibogaine is found in the Tamphananth iboga, which is an ethnobotanical plant, and it's found in West and Central Africa, particularly Cameroon. And it's widely used in religious ceremonies in this region, where people who are joining the religion, initiates, will chew the root bark of the Iboga during overnight rituals, which causes these initiates to have visionary experiences. People report going back to their childhood, things of this nature. However, because it creates some very adverse effects, such as nausea and even death in some cases, or just what people would explain as like a bad trip, it's rarely used recreationally. It's almost exclusively used in religious or medicinal applications. So the tea iboga plant was first introduced to the West in 1864. The plant was brought to France along with tales of its ritual use. And Ibogaine was first crystallized from the root bark in 1901. And from 1939 to 1970, Ibogaine was used in France medicinally as a neuromuscular stimulant. That's the term that they use for treatment of a wide range of conditions such as fatigue, depression, and infections. Today, the iboga root bark is used clinically in Mexico for treatment of substance use disorders. And I'll share with you in just a second that there used to be Ibogaine treatment providers in the United States. However, that's no longer the case because Ibogaine is a scheduled drug. And you really can't talk about Ibogaine without mentioning Howard Lotzoff. So Howard Lotzoff was a heroin addict who was part of a group in the 1960s who experimented with different psychedelic drugs. And Lotzoff was in this group with about 19 other members when they all took Ibogaine together. So out of the 20 individuals, 7 were dependent on heroin, including Lotzoff. But after taking Ibogaine, 5 out of the 7 claimed that they had been cured of their addiction. So by cured of their addiction, they mean that they didn't experience withdrawal symptoms despite not taking the drug or experience cravings. Because of this reportedly miraculous effect of Ibogaine, Lotzoff, despite the fact that he was just a layperson, he didn't have any medical or scientific training, basically made it his life's mission to proselytize about the wonders of Ibogaine. Although he was not ever successful in convincing a pharmaceutical company to take on this project, he did patent Ibogaine for the use of treating substance use disorders of a number of different substances. And he did convince the researcher Stanley Glick to research Ibogaine in rodents. And Stanley Glick's results were quite amazing, I feel. They supported many of the claims of Howard Lotzoff that Ibogaine did in fact have anti-addictive properties. So just briefly, I want to share with you the synthesis of Ibogaine. So the first total synthesis was published in 1966. However, this simplified total synthesis of Ibogaine that was published in 2012 shows that Ibogaine is reached at a 9.8% overall yield. Now, it's still a mystery how Ibogaine causes its effects because there's no clear receptor preference. So Ibogaine and most of its analogs bind weakly to many different targets. However, it's unique in its hallucinogenic properties because it's not caused by action with the 5-HT2A receptor, unlike many others like LSD, mescaline, and psilocybin. Additionally, the most promising cause for its anti-addictive properties is its interaction with nicotinic acetylcholine receptors. So as you can see from the table, Ibogaine is a non-competitive antagonist at several nicotinic acetylcholine receptors, including alpha-3 beta-4. And the reason why I'm saying that it's not specific is because it binds to all these different receptors, but in terms of reacting with nicotinic acetylcholine receptors, it has a preference for alpha-3 beta-4 over others. So as I hinted to earlier, Ibogaine, although it has very promising anti-addictive effects in rodents, it's extremely controversial due to its life-threatening effects. So in rodents, Ibogaine causes convulsions, nervous behavior, neurotoxicity, even limb paralysis. And in clinical studies, the main adverse effects were nausea and cardiovascular problems. In terms of its psychedelic properties, Ibogaine causes you to think back on your past, particularly into your childhood. It causes people to recall traumatic childhood experiences, and also can allow drug addicts to perceive their addiction in a different way. So people describe this as sort of like a dreamlike state. And this is actually why Howard Lotzoff felt that Ibogaine had anti-addictive properties. He didn't believe that it was necessarily anything biological that was happening. He felt that when he had his experience with Ibogaine, it gave him just a different perspective on his addiction. It gave him a different perspective on his life and where he was going. And this basically like opened his eyes and made him change his trajectory. This is what he reports. So there was a recent study of 22 Ibogaine users in Brazil, and they specifically said that they experienced 3 different stages of Ibogaine high. So the first stage is the first 4 to 8 hours, and it consisted of intense emotional and perceptual feelings. The effects then would decrease in the following 8 to 20 hours, which is the second phase. And then in an additional 1 to 3 days, they would be back to their normal consciousness, which is the third phase. So the next compound that I want to share with you is called 18-MC. So 18-MC is a synthetic derivative of Ibogaine, and it was developed by Stanley Glick, the man and researcher I told you about earlier. He had tested Ibogaine in rodents and saw that it had these anti-addictive properties. However, he also noticed its toxicity. So his goal was to create synthetic derivatives of Ibogaine that maintained its anti-addictive properties, but eliminated or lessened their toxicity. And he was successful in creating a number of different analogs. However, the one I'm sharing with you today is called 18-MC. So it has less toxicity, but it also has been shown to inhibit morphine and cocaine self-administration in rodents. And it lacks some of the negative side effects, such as tremors and neurotoxicity associated with Ibogaine. And 18-MC is currently in clinical trials for the treatment of nicotine addiction. The other structure that I have on this slide is noribogaine, which is an active metabolite of Ibogaine. And it also has been shown to have these anti-addictive properties. And the reason why I've included noribogaine and why I think I'm not going to go full into detail on it, but why I think it's interesting is because it's been suggested that the long-acting nature of Ibogaine's anti-addictive properties is due largely in part to the fact that its active metabolite in the body, noribogaine, is also has also these anti-addictive properties. It's also active. So I thought that was kind of interesting. So the last compound that I want to share with you is aristoquinoline. Aristoquinoline is a natural alkaloid found in Aristotelia chilensis, which is found in South America, also called the maqui tree. And it's found in the leaves of this tree, and you can see the structure of it here. So recently, in 2019, a study by Arias and others suggested that aristoquinoline is a somewhat selective antagonist of alpha-3 beta-4. As you can see from this table, it has increased affinity for alpha-3 beta-4 over alpha-4 beta-2 and alpha-7. It's an up-and-coming compound in terms of its anti-addictive properties. Although it hasn't been tested biologically yet, there is a great suggestion and a hypothesis is that because it is also a selective alpha-3 beta-4 antagonist, it will have similar anti-addictive properties as the others that we've talked about, such as Ibogaine and 18-MC. So in conclusion, the natural alkaloid Ibogaine is a double-edged sword. So on the one hand, it produces this fascination amongst people because of its psychedelic properties, and because it is kind of touted by Lotzoff and others as sort of an anti-addiction magic bullet. However, it also produces dread and fear because of its unpredictable side effects and its lethality. So one of the big issues with Ibogaine is that when it's been introduced into clinical trials, particularly in France, it's caused the death of the participants, which is, rightfully scaring off people from continuing to investigate it in humans. So Ibogaine, and also to some extent the metabolite noribogaine, have properties that lessen drug-seeking behavior. They mitigate dependence on drugs, many drugs of abuse, including, I focused on this talk, psychostimulants. But unfortunately, there's still a heavy price to pay in the fact that they have so much adverse side effects, such as cardiovascular damage, neurotoxicity, and death, as I mentioned. So this search for a compound that could retain the positive aspects of Ibogaine, but get rid of the neurotoxicity, has led to 18-MC, which is a compound with tremendous potential, as I see it, because it also inhibits the self-administration of many drugs of abuse in rodents. And it's, like I said, currently in clinical trials. And of course, we can't end this without mentioning again Howard Lotzoff, his tremendous effort in bringing Ibogaine to the public eye, getting the first research done on it, and no longer these sort of just circles of people psychedelics, but actually people testing them in the lab. He's put a tremendous effort into putting Ibogaine and other alpha-3 beta-4 antagonists onto the scene. So with that, I will take any questions.